अगर हम लोग म्यूटेशन का बात करें सर, तो म्यूटेशन क्या है सर? इट इज एन इनहेरिटेबल चेंज इन एन ऑर्गेनिज्म. किसी भी ऑर्गेनिज्म में जो एक इनहेरिटेबल चेंज होता है, that particular inheritable change is known as mutation. Now whenever we talk about studying mutation, we're gonna study it under different headings. Okay, क्या क्या sir? So we'll see that the mutations can be divided into two different types. One is called the gene mutation and one is called the chromosomal mutation. Now if I'm talking about gene mutation, we're gonna study mutations like your silent mutation. You're gonna study mutations like missense mutation. We're gonna study mutations like nonsense mutation, your substitution mutation, and at last the frame shift mutation. On the other hand, if we are talking about your chromosomal mutation, we can divide chromosomal mutation into two different types. And what are these two different types we have? So the first type is called chromosomal aberrations. ठीक है, एक टाइप को हम लोग क्या बोलते हैं? chromosomal aberrations बोलते हैं. Now what is chromosomal aberrations? तो chromosomal aberrations is something when there is a change in structure. There is change in structure of an organism, but the number remains the same. And on the other hand, we talk about a change in chromosome number. Now if I'm talking about change in chromosome number, we're gonna come across two different things. We're gonna come across euploidy, where entire set of chromosomes change, and aneuploidy, where only few chromosome number changes. Now in chromosomal aberrations, we're gonna study things like deletion. We're gonna study things like duplication. We're gonna talk about translocation. We're gonna talk about inversion. In euploidy, we will have two different types. One is called the haploidy, because if a diploid organism is there, it is changed into haploid structure, then it's a mutation. And polyploidy. Under polyploidy, we're gonna study two different types. We're gonna study autopolyploidy and we're gonna study allopolyploidy. So these are the things which we're gonna study in details. लिख लीजिए. Now see, before going to gene mutation, we need to understand one thing, that we have something called DNA, and from DNA, what will happen? The RNA will form. So if there is any change in nucleotide here, that will be reflected. It will be reflected here. It will be reflected here. So we are not gonna write the sequence of the DNA. We are gonna just write the sequence of your RNA. If I am changing something here, there, you have to assume that the change has been taken place in DNA. Now, for example, if you remember, we understand one simple fact, that your RNA lead to the formation of protein. Okay, and when we are talking about RNA lead to the formation of protein, we see they act as triplet codon, with the help of which the information is passed. So we are writing down some triplet codon here. So this is your particular one sequence. Now if you look at this sequence, you will see that AUG is coding for methionine, CCC is coding for proline, GUG will code for valine. If you are talking about ACG, it is coding for threonine. GCA is coding for alanine. UUA is coding for leucine, and ACG is again coding for threonine. So this is a basic mRNA sequence which we are taking, and with the help of this structure we're going to study all different kind of mutations. So the first mutation which we have is called silent mutation. Now what happens in case of silent mutation, we'll see that. So we'll just demonstrate your silent mutation. AUG, CCC, GUG, ACG. Now GC. In place of A, what do we see? G is coming. Okay, so what we're gonna do, we're gonna change the color here so that we can identify where is the particular change which is taking place. So we have GC, G. Okay, हम लोग को क्या मिल रहा है? GC, G का formation देखने को मिल रहा है सर. Let me change it with red color. Better. Now, after that we have your UUA. So we have UUA and we have ACG. Now this will code for methionine, this will code for proline, this will code for valine, this will code for threonine. Now GCG do not code for your particular alanine. Okay, no, sorry. GCG also code for alanine only. So what you're gonna get is you're gonna get alanine, you're gonna get leucine, and you're gonna get threonine. So what does silent mutation means? That there will be a change in nucleotide sequence, but the effect will be not seen in phenotype. Is it clear? See, from GCA the sequence has been changed to GCG, but still there is no effect in the particular protein sequence. Now after this we'll study another type of mutation. The second type is known as the missense mutation. Now talking about missense mutation, let us write down the sequence again. We have AUG, we have CCC, we have your we have your GUC. Okay, one second. GUC. Fine. We have your GUG. So we'll do what? We're gonna change this sequence from GUG to your AUG. So what is changing? In place of G, you are getting A. So I will write down GUG. Oh, sorry, I have written the same. So we're gonna get what? We're gonna get AUG. Now what next? So next, if we're gonna see, we have ACG, we have GCA, we have UUA, and we have ACG. Now if you see, this will code for methionine, this will code for proline. Now in place of coding for valine, we are going to get here what? We are going to get methionine, and this all will remain same. So missense mutation says what? That due to change in nucleotide, even amino acid changes. Okay, and this kind of mutation will be called your missense mutation. Is it clear? Clear है बाबू ये सब? तो देखो स्टैंडर्ड वाला फिर से नीचे लेके आ गए. अभी इसी के बेसिस पे आगे देखते हैं. तो हम लोग क्या देखेंगे अभी? नेक्स्ट म्यूटेशन इज कॉल्ड योर नॉनसेंस म्यूटेशन. What happens in case of nonsense mutation? Now see here. For example, this remains same: AUG, CCC, GUG, ACG, GCA, U. Now this U is converted to A, and the sequence is now UAA and ACG. Now what we'll see, this AUG will code for methionine, this will code for proline, this will code for valine, threonine, alanine, but this is a stop codon. So the translation will stop here. So what we see here in nonsense mutation, that in this, due to change in nucleotide, the codon is converted into a stop codon, and as a result, translation is terminated and lead to incomplete प्रोटीन. ठीक है, वो आपको इनकम्प्लीट प्रोटीन का फॉर्मेशन कराता है. क्लियर है वो? अभी देखिए, इसके अलावा नेक्स्ट म्यूटेशन जो हम लोगों को देखने को मिलता है वो क्या है सर? तो वी विल सी समथिंग कॉल्ड योर substitution mutation. So what do we see here is, in this, one nucleotide is exchanged or changed, not exchanged basically, is changed by another nucleotide. So what do we see here is, the substitution mutation is of two different types. One is called transition type. So what happens in transition? We have something called purine. If the purines are replaced by purine, means you have guanine, and if it is interchanged with adenine, then this kind of mutation is known as transition. And at the same time, if pyrimidine is replaced by pyrimidine, means your cytosine is interconvertible or inter-exchanged with thymine. Now another is called transversion. Okay, in case of transversion, what we see, in this purine is changed by pyrimidine. So basically we want to say that if adenine is there, it will be replaced by your particular thymine or cytosine, and if guanine is there, it will be replaced by your particular thymine and cytosine. Now see, if we are talking about frame shift mutation, हम लोग यहाँ पर क्या देखेंगे कि this can take place by two different methods. One is called deletion, okay, and another thing is called addition. Now talking about deletion and addition, let us write down a sequence, then understand this thing. For example, you have a sequence called AUG, you have CCC, you have GUC, you have CGA, and you have UAC. So this is a sequence, and what we are saying here is, for example, if your guanine is getting deleted, so the new sequence will be AUG, CCC. Now this got deleted, so you will be UCC, GAU, UA. So due to deletion of nucleotide, what is happening due to deletion of nucleotide? The amino acid are changed from point of deletion. From point of deletion. Let us see सही है कि नहीं जो हम लोगों ने लिखा. AUG will code for methionine. Both of them will code for methionine. No problem for that because deletion is here. CCC will code for proline. No tension for this one. Now GUC is coding for your valine. Okay, this is coding for your valine. Your CGA is coding for your arginine. And your UAC is coding for your tyrosine. Now because of deletion you got a sequence called UCC. Now UCC will code for serine. Okay, not for valine. Now GAU will code for valine, not for arginine. And because here the particular codon is incomplete, there will be no amino acid which will be forming. So due to deletion, from the point of deletion, all the amino acid will change. The second type which is here is addition. Now in case of addition, let us see the same standard sequence and try to add something. For example, AUG, CCC. Now we are adding a molecule here and you are getting GGU, CCG, AUA and C. So from the point of addition, the sequence has been changed. So this will code for methionine. No problem with that. CCC will code for proline. No problem here also. GGU will code for glycine in place of valine. CCG will code for proline in place of your arginine. AUA will code for isoleucine. So you can see that from the point of addition, the type of amino acid changes. Okay, so this is your frame shift mutation. Write down. Which questions देखेगा हम लोग? First, like in which condition does the frame do not shift? So I'll give you certain options. In which condition the frame will not shift? One nucleotide is deleted or added. If your one nucleotide is deleted or added, or your two nucleotide is deleted or added, and third one, your three nucleotide is deleted or added, and fourth one is none of the above. Out of this, which condition the frame will not shift? The answer will be your when three nucleotides are added or deleted. For example, AUG, CUC, GUC, GGG. Okay. Now, if you delete one C, what will happen? The new sequence will be UG, UCG. If you delete both of them, you will get CGU. Now, if you delete this also, the sequence will remain the same. It will be AUG followed by GUC followed by GGG. The entire one triplet codon will be replaced and there will be no change in the frame here. Now, this kind is only possible when you have three nucleotide getting deleted. There is another common question which comes from this particular place, that if there is a RNA with 1000 nitrogen bases and codes for 333 amino acids. If 901th nucleotide is deleted, then how many amino acid will get affected. Now for this question you need to remember one thing. We have already discussed that the effect will be only seen from the point of deletion. So the deletion is on the 901th place. So 33 amino acid will get affected. Why we are saying this? Because the initial 900 will form your 300 amino acid, and from 901 your 33 amino acids are left. So there will be total 33 amino acid whose sequence will be affected. लिख लीजिए. Next हम लोग क्या देखेंगे? We will talk about chromosomal mutation. Now if I am talking about chromosomal mutation, we have discussed that under two headings. One is chromosomal structures change, and one is with the chromosome number change. So first let us deal with the structure thing. So if we see, our chromosome structure can be changed due to deletion. Now if I am talking about deletion, what happens here? That some portion of a chromosome get deleted. Now deletion can be taking place with two different methods. One, the deletion can be terminal in nature. For example, if we have a chromosome with 1, 2, 3, 4 and 5 genes, and the deletion is such that you are going to get something like this. For example, 2, 3, 4 and 5. What happened? The gene number 1 is getting deleted, and it can be interstitial. In this, what will happen? The deletion will be from middle. It will be something like this: 1, 2, 3 and 5. Okay, so if the deletion is like this, then we call it your interstitial type. Now इसको R को ठीक कर दो. Now see here, what will be the problem? Obviously the gene which will be deleted will be not able to be expressed, and if the gene is not expressed, obviously that particular protein will be not present, sir. After this we have duplication. Now in duplication what happened? A portion of chromosome get duplicated. Now before understanding this, let me give you an example of this deletion part. We have a disease called Cry-do-chat, which is because of the deletion of, which is due to a deletion in long arm of chromosome number 5. Okay, this is something which can be asked in question. Now duplication, for example, if we take this as the standard and we see something like this, that you have 1, 2, 2, 3, 4 and 5. So if you see, this portion is getting duplicated. And what will be the problem with this? For example, if the gene for insulin get duplicated, you will have more amount of insulin which will be synthesized in your body, and that can lead to something known as your diabetes. Now the third type which we see here is called your translocation. Now translocation, what happened? Here a portion of the chromosome is transferred from one chromosome to another chromosome. For example, when portion of long arm of chromosome number 9. Now chromosome number 9 is also known as Philadelphia chromosome of chromosome number 9 is transferred to chromosome number 22, it lead to your leukemia. Okay, it lead to your leukemia, your chronic myelogenous leukemia, type of cancer. इसको लिख लो. Last type of your chromosomal variations is your inversion. Now inversion, what happened? Your genes will be rearranged. Genes are rearranged in a chromosome due to switching. So what you will have? You will have two different types. You have something called paracentric and you have something called pericentric. Now for example, if you have a chromosome here, 1, 2, 3, 4 and 5G, and if the switch is something like this, that your particular chromosome are exchanging the particular genes in such a manner that the centromere is not involved, means you're gonna get something like this. You'll have 1, 2, 3, 5 and 4. So here centromere is not involved. So if this is the condition, we call it paracentric. In pericentric, what happens? For example, something like this. You have 1, 3, 2, 4 and 5. Your centromere is involved in the rearrangement. So that is called your pericentric inversion.